presentation from Professor Michael Kuhns, who is an associate professor in immunobiology in the College of Medicine here in Tucson. Michael got his BS degree in biochemistry right here in Tucson and went from there on to Jim Allison's laboratory at UC Berkeley, where he studied CTLA4, laid the groundwork for what became fantastic therapies and a Nobel Prize for the Allison Laboratory. He then went from Berkeley all the way down the peninsula to Stanford with Mark Davis, where he continued his work on understanding T-cell activation and T-cell receptor complexes. We recruited him back here to Tucson in 2010, where he joined immunobiology. He's continued his studies to make both give fundamental insights to T-cell receptors, and as well as to develop translational components in CAR-T, which I think we'll probably hear some about that momentarily. So Mike, the screen is yours.
All right, thanks, Bill. I appreciate the introduction. I just want to clarify that the really groundbreaking work in Jim's lab was done right when I joined, but I didn't do that. I didn't have the pleasure of doing that work, even though I did work on CTLA4, but I appreciate the introduction.
I'm sure you influenced it.
All right, well, so I know we have a broad audience today. I appreciate the opportunity to share our work with you. And what I want to do, because I'm an unabashed basic research scientist, is give you an introduction to the immunology that you need to know to understand what we're doing, as well as some of our basic work, which is inspiring and informing our biomimetic engineering efforts. And then I'll get into that new technology. And I just have to briefly add a disclosure that I have an interest in module therapeutics with the technology that I'll talk about today. All right, so by way of introduction, all of us as vertebrates have an innate and adaptive immune compartment of our immune systems. The innate cells are sentinels. They provide you with the first line of defense. And the adaptive cells include the T cells and B cells that we've heard about today, which provide you with long-lived protection against microbial infections or vaccines, or even neoplastic transformation. Now, a key thing for all of us to be here happy and healthy is that these cells have to talk to each other. And so my lab, along with a number of other immunology labs around the country, are concerned with understanding how productive immunity emerges from these conversations. And the way that my group is going about trying to contribute to our understanding of these processes is to study the molecular machines that line up at the interface between an antigen presenting cell and a T cell to relay specific information across the T cell membrane into the T cell to inform its fate decisions. All right, so what do you need to know? This is the key determining event for T cell activation. The top molecule you see here in white and magenta is the T cell receptor bound to a peptide antigen here in yellow, presented by a major histocompatibility complex or MHC molecule. And that peptide could be derived from a self-protein, or it could be derived from a microbial protein or a vaccine or a neoantigen. And then the duration of this interaction between the TCR and this peptide MHC will dictate the nature of the information that goes up inside of the T cell. Now, the TCR itself lacks an appreciable intracellular domain. It cannot signal, and instead it associates with the CD3 delta epsilon, gamma epsilon, and zeta zeta signaling modules, which have signaling motifs called ITAMs. And they connect this receptor complex to the intracellular signaling machinery. Now, this complex has 10 of these ITAMs, but no intrinsic kinase activity. And so dogma has it, for those of you who have taken immunology, that when a TCR CD3 complex binds a peptide MHC, a co-receptor such as CD4 shown here or CD8 that's not shown here will recruit a non-covalently associated Src kinase, LCK, to phosphorylate the ITAMs and initiate signaling. Now, about 10 years ago, when I was starting my lab, this was the dominant paradigm in the field. based on two crystal structures of super physiological interactions between CD4 and peptide MHC. People thought that there might be some sort of V-type orientation of TCR peptide MHC with CD4. And for us, that created some conceptual problems, both in terms of understanding the very weak interactions between the TCR and the peptide MHC, and the really weak interactions between CD4 and class 2, so weak they can't be measured, they can only be guesstimated, as well as this distance here between the kinase and the ITAM. So taken together, we were wondering how this could create a good local concentration for a sufficient amount of time to really get all this stuff to generate a signal. And so we hypothesized that perhaps this was TCR signaling was more akin to cytokine signaling, where we know in a basic model, for example, with the IL-4 receptor alpha subunit, that when it binds its ligand IL-4, that creates a composite docking surface for the common gamma chain, a degenerate subunit that otherwise has no appreciable affinity for IL-4, but it can bind that composite docking surface to form this higher order assembly. And what that ends up doing on the inside of the cell is recruiting the associated Janus kinases to initiate JAK-STAT signaling. And so we thought, well, maybe akin to that model, TCR docking to peptide MHC and coincident binding of CD4 and a V-like arch might be an intermediate step. But really what would happen is that the TCR CD3 peptide MHC axis might create a composite docking surface for this degenerate CD4 chain to bring the Src kinase LCK in close proximity to these ITAMs and initiate signaling. And this was kind of nice because it gives you two clear, distinct models with testable predictions. And I'm just going to walk you through one prediction, this issue of distance here between CD4 and the TCR complex. If you use a technique called FRET, which I'll explain in a second, these regions should be too far apart to measure an interaction, whereas in this more compact assembly, these things should be close enough to measure an interaction. And so FRET is a technique where if you have a donor and an acceptor in a close spatial relationship with aligned dipoles in a radiationless manner, after exciting the donor, you can get energy transfer to the acceptor, which will emit that energy as light. And that the efficiency with which that occurs is proportional to the inverse 6 power of the distance between those dipoles. So if we put a donor on the TCR complex and an acceptor on CD4 and look for FRET, we would expect in this model that we shouldn't see any interactions. And the way we test this is, in fact, to ablate the acceptor and look for the donor to unquench or get brighter and calculate that FRET efficiency by this formula here. And the way we measure that is by total internal reflection fluorescence microscopy, where we ablate the acceptor as shown here. And then you can see the donor over here blinking on in pseudo color or here in grayscale. In this fretting cell at the membrane. And so essentially, if we looked at TCR, CD3 complex, and CD4 in an unengaged state, we see no FRET. This is equivalent to a negative control that I'm not showing here. And if we then provide the ligand and engage this on a mobile lipid bilayer, we get a nice increase in FRET. And so that data would be consistent with this model instead of this. And Caleb Glassman in my lab tested a variety of different predictions of these different models. And in each case, in these two papers, we found that the data were more consistent with this model than this model. And so we think that this is the final assembly of how all this works. So how all this might be useful. Joe, in the first talk, introduced this idea of CAR T cells or CAR T cell therapy, these so-called living drugs. And so we thought, well, the work that we do is certainly applicable to this sort of approach of synthetic receptor engineering. So this is what a CAR T cell idea looks like. You have a tumor cell with a tumor ligand or antigen, and then you have a single chain FV from an antibody binding that tumor ligand, and it's hooked up to a string of intracellular signaling motifs expressed in a cytotoxic T cell, CD8 T cell. It should redirect the T cell to kill the target cell. Now we heard about the cytokine release syndrome. And another problem, and one reason why these CAR T cells are generally not curative, is because they are much less sensitive than their native counterparts, somewhere between 100,000-fold less sensitive. And so very often tumors come back that have reduced tumor antigen expression, and they've just escaped the CAR T cells. At least that's part of the problem. Now, why might this be? Well, if we look at the blueprint that evolution has come up with for directing T cell activity, you can see that you have a receptor complex that's held apart from this co-receptor and kinase until the appropriate ligand is encountered. And I can tell you that this assembly can signal in response to one peptide MHC and kill a target in response to 3 peptide MHC. By comparison, the single chain CAR, what we'll call a one module CAR, as opposed to the TCR, CD3, and CD4 5 module assembly here, this one module assembly was developed or designed in the early 90s, when we understood that a receptor would bind its ligand and have signaling motifs, and everyone just assumed that the information got across the membrane by clustering. And so that was the extent to which this was designed, was really relying on clustering. And there's no way to bring kinases in to modify these motifs, these intracellular motifs here, to initiate signaling. So what was our idea? It was basically to build a CAR by biomimicry, to make something that would mimic the native 5-module system. So we're going to call that a 5-module CAR. And this idea of biomimicry is one that's taken hold in chemistry, material sciences, and engineering. It's sort of lagging behind in the cell and molecular biology, but we're hoping to bring that up to speed at some level. And so what have we designed? What we decided to do at first was turn T cell antigen recognition on its head by making the peptide MHC the receptor module. So we engineered what we call a chimeric receptor module that's a fusion of the peptide MHC ectodomain with key elements of the TCR transmembrane intracellular domain, such that it will assemble into a complex with the CD3 signaling modules. And then we also engineered what we call a surrogate co-receptor module, which is basically based on the ectodomain of CD80 fused to LCK. And we chose this molecule because imaging experiments from our lab and others suggested it comes very close to the TCR. Well, its ligand would. So it should come in very close proximity to this. And the notion would be that this 5-module assembly would mirror the form and function of its native counterparts. So what might this be good for? You can imagine if you have a pathogenic T cell that is mediating autoimmunity against some self-tissue by recognition of a self-peptide MHC, then by arming a CTL with one of these 5-module CARs, based on that self-peptide MHC, you could actually target the pathogenic T cell to kill that pathogenic T cell. All right, so is this superior to 1M CARs? We tested this head-to-head. We made a 1M CAR with a self-peptide MHC to target a T cell population, and then we used the same peptide MHC to target in our 5M CAR to target that T cell population. And when we compare them head-to-head in an in vitro killing assay, we can say if we have the wrong peptide in the 1M CAR, we don't get any killing of our T cell targets. With the right peptide, we do get 20-30% killing. And then with our 5M CARs, again, the control population doesn't kill. And with the right peptide, we get much more robust killing, somewhere around 70%. So we're getting better killing in our 5-module system. Now, can these 5M CARs kill autoimmune T cells in vivo? So we collaborated with my friend Tom Sirwald at the Joslin Diabetes Center in Boston, who had a nice system where you can transplant an autoimmune T cell population into non-skid mice, and then reliably 9 to 11 days later, these mice get type 1 diabetes. And so we decided to do a very rigorous test of hunting them down 7 days later with our 5M CAR populations. And the reason why we picked day 7 is because if you look at the islets of just one of these non-skid mice with no autoimmune T cells, this is what a healthy islet looks like. Even by day 5, you can see insulitis, and by day 7, the integrity of these islets is falling apart as those autoimmune T cells are in there mediating destruction. And just a couple days later, these mice will get sick. So if we look in this model, untreated mice get sick, as I said, between 9, 11 days. If they're treated with a control group of a 5M CAR CTLs with the wrong peptide in our CAR, they also get sick with the same kinetics. But if they have the right peptide to target the autoimmune T cell population, only 1 in 9 mice got sick in this experiment. And so we were able to rescue the majority of these mice from getting diabetes. And if you want to know what's happening in vivo in these mice, these are flow cytometry plots of the pancreas of mice, either at day 7, 8, 9, 10, 15, 36, either the day before transfer, the day after transfer, and thereafter. And we're looking at our 5M CAR T cell population here and the autoimmune T cell population here. And so what you can see is that 1 day after transfer, the CAR T cells show up in the pancreas in both groups. But the control group has a higher frequency of the autoimmune target T cells than the specific group, the treatment group. And by day 10, it's pretty obvious that there's a big expansion of the autoimmune T cells. This is when these mice are really getting sick. But in our specific group, they're really not expanding, they're diminishing. And by day 15, in the treatment group, that autoimmune T cell population is completely wiped out, it's gone, whereas these mice have to be euthanized because they're diabetic. And then this response is durable out to day 36. There's no recovery of the autoimmune target population, and instead we see a remaining population of the CAR T cells in these mice. So we also asked, can these be used to treat or prevent spontaneous type 1 diabetes and replete T cell replete mice, so full NOD mice? And these NOD mice, they get autoimmune priming of the T cells very early in life. And so what we decided to do was either inject a control population or a population of 5M CAR CTLs with 4 known different specificities for initiating type 1 diabetes in the NOD mouse. And then we track these mice by urine glucose weekly out to day 315 to monitor diabetes. And what we can see in the untreated or control groups is that the majority of these mice, 85–90%, got diabetes out at 315 days of age, whereas in the treated group, just under half of the mice got type 1 diabetes. So it wasn't perfect. We didn't cure all of them. There are some technical issues here, but we think this is very promising. And so just in conclusion, we think that this first generation 5M CAR approach can be used to redirect CTLs to eliminate these autoimmune T cells in a clonotypic TCR specific manner. And we think that this then shows promise for treating a variety of different autoimmune diseases that are T cell mediated. And we think that there are ways in which these could be used to treat T cell lymphomas, such as the PTCLs that Stefan was telling us about. And with that, I just want to thank people in the lab who have done all this work in collaboration with Tom Sirwald and Martin at the Joslin Diabetes Center. And Shio did a lot of the 5-MCAR work, shuttling back and forth between the two labs. And this group of people did a lot of the basic science that I told you about, as well as some of the 5-MCAR work. Funding sources, and thank you very much. I'll take questions.
Thank you, Mike. Exciting as always to see your progress. So we have time for some questions here. We don't have any in the question box just yet. So that leaves me plenty of room to start peppering away. So one question I have is do you see a way for this to actually treat diabetes though? Because this occurs at such an early stage in patients that get this disease, the type 1 diabetes.
Right. Well, it's a good question. So I think the way to think about that is that usually patients get, what will happen is you'll see two autoantibodies prior to the first sort of diabetic episode. And so those autoantibodies are probably driven by autoimmune CD4 T-cell responses. Those antigens almost certainly involve this dominant insulin antigen and perhaps a couple others, but it's probably very limited. And so that would be a good population to think about trying to treat before in this honeymoon phase, basically is what they call it, before then they have full islet destruction, because those individuals still have islets at that point in time. And so that would be the place we would start looking. Yeah.
All right. So it'd be sort of one of the many early diagnostics we do on babies, I guess, to see if there would be a treatment option. So that'd be very exciting.
Possibly, yeah. I think you'd have to be careful in clinical trials who you selected, and we'd have to have more knowledgeable clinical partners than me.
Well, we have them around. So we have a question in the chat box now from Souvenir Chado. Have you looked into the effects of your compounds, your he's calling it stuff, I'll call it T-cell modifications on endosomal TLR activation? And does it involve in its involvement in autoimmunity?
The short answer is we have not looked at that, no.
Okay. Yeah, I don't have any comment on that either. Selena, go ahead.
Yeah, thanks, Mike, for a great talk. I was wondering, could you combine? Are you thinking about combining the traditional, what we call M1 with or 1M or 5M? I don't remember exactly the terms they use, but could you potentiate by combining both interactions? So
if I understand your question right, you're asking if, for example, we could combine some of our other modules with the more classic CAR design.
Yeah.
Right. So that's a great question. And we've looked at that and we've seen marginal effects. And so a variety of people have looked at the classic CARs, and, they don't have a full complement of ITAMs, and there are other issues. And it also depends on what which generation or version of intracellular motifs people are using. We think that the surrogate co-receptor idea could in fact be useful for enhancing the function through the classic CARs. That's the short answer. We've looked at it, we see some impact, but they're not as good as the system we built.
Okay, great, thanks.
Okay, we have a comment from Ron Lynch on the diabetes component. Who notes that there's a strong genetic component in type 1 diabetes. So the focus could simply be on susceptible families, which would be outstanding.
Yeah, Ron, that's a great comment. I think there are people who are in high-risk groups, and that would certainly be the group of people we would want to recruit from for any kind of trials.
I have another question for you, Mike. So everything that you've done so far with this these fantastic new tools is aimed at T cells. I'm wondering if you're thinking about how you can turn it on its head and go after solid tumors, which is sort of the original CAR T target.
Right. So people are trying a variety of different approaches with these CARs and the ligand modules. So at about the same time our design came out, another group was using an antibody in what they call a true C or a truck type thing, where they hang a anti-tumor antigen FV off of one of the subunits of the TCR complex. They don't have a co-receptor design, but you could imagine you could take one of our designs and combine it with that. I think the binding kinetics wouldn't be quite right there still for the TCR complex. So you might imagine using affinity tuned FVs as a receptor module in this approach. And I think people are thinking about and trying these different things. I'm also sort of biased actually to the T engager route. And so we have ideas and efforts at sort of more biomimetic T engager approaches that I think in the end would be a lot better than gene therapy.
Yeah. Indeed, if you didn't have to actually edit the genome, that would be a good thing.
Yeah, I would prefer an injectable.
Okay. Seeing, I think we're out of questions. No, there's one more here from Souvenir Tashado. Have you looked into using ITIM instead of ITAM?
We haven't, I guess. I'm not clear on the sort of the idea. What would that give us? What advantage that would give us? The short answer is we haven't done that yet, or we haven't thought about it actually. But I'll have to ponder it more. So I appreciate the question.
Yeah. Well, maybe we'll get him to track you down and have a further discussion on it.
Right.
Okay. Well, thank you, Mike. I think we'll move on to the next speaker.